afternoon to everyone here, and I'd like to thank the organizers for the invitation and also for all of you for being here. I'm Hari Nair. I'm a clinical chemistry fellow at the University of Washington, and I'm going to talk about some of the recent work we did there with respect to developing a clinical assay for relative quantitation of diverse classes of lipids using LCMS. So, so the goals of the work is that we wanted to have develop an LCMS assay to predict myocardial infarction in patients that are presented to ER with acute fibrillation. So there are two things that we wanted to accomplish. We wanted to do a targeted discovery of lipids that are implicated in this process, and we wanted to use an assay for routine analysis of these lipids. The specimen that we want to look at are plasma erythrocytes and HDL from these patients. So we have the Department of the University of Washington Medical Center has been collecting a large number of, we have a cache of large number of samples. We've been collecting samples for about 15 years from patients that have been brought to the ER for acute fibrillation, and we can look at all those data and see if we can find a predictor to a myocardial infarction. And so we need a method for that, and a method that can be easily adapted to clinical lab. So that was the challenge. So the challenge is because our hypothesis wanted us to look at a variety of different, chemically different lipids, such as phosphatidylcholine, sphingomyelins, which are generally highly nonpolar, and then we wanted to look at some of the polar ones, like the glycated ceramides. So in order for this to work in our lab and to be able to analyze large number of samples, thousands upon thousands, we needed to have them all in a single workflow, and we wanted to look at plasma, RBC, and HDL. So why do we want to measure these particular lipids? Because this is the only biology slide I have. So why do we want to look at these lipids? Because sphingomyelins are implicated in, they are independent predictors of coronary artery disease and implicated in atherosclerosis. And ceramides are the backbone of sphingomyelins, and they affect, through their second messenger role, affect atherosclerotic changes. These are known facts. And phosphatidylcholines are key components of lipoproteins, and so we wanted to look at them as well. So the question is, can we use these lipids to predict myocardial events? And more importantly for the hospital, the question is, can we use these markers to be able to discharge or admit the right patient? Because a lot of money and a lot of patient care rides on this particular question. And so this question was the driver to this particular work. So switching back to analytical aspects, so our goal was to develop an assay that fits our clinical lab constraints and resources. And so we wanted to have a workflow that has a single extraction step, a single separation step, and be able to be used in a triple quad, because triple quads are the most dominant platforms in clinical labs. And like I mentioned, we wanted to assess the performance in plasma, erythrocytes, and HDL. And so, as I mentioned, one of our goals is the discovery. So we wanted to have an expanded, as big a target list as possible to look at. And for this, we compiled a large list of isobaric parents from databases such as lipid maps, and we used that as our targeted database. And the most important thing, whether to go or not, question is the cost of the assay, because we have to look at thousands of samples, and we do this all on the side of actual clinical running. So the cost is extremely important, and so we wanted to cut costs. We wanted to use a single internal standard, if possible, to normalize the signals of all the peaks that we'll be detecting. So, in other words, this is what we wanted to do. We wanted to analyze all the lipids in a single multiplexed assay, get everyone together cheaply and in one place. So, are there methods, does methods exist that can be used to do this? Yes, there is a lot of literature out there where many of these lipids can be characterized, and particularly this, I'm bringing to notice this particular assay developed by Merrill and co-workers and adapted by manufacturers of some of the lipids, such as Avanti polar lipids, to QC the batches of their lipids, and also recommended by lipid maps. So if you look at this assay, this is a very good assay for characterizing a variety of lipids. But as you can see, it also has large number of steps. So, for example, one of the steps is a 12-hour incubation, followed by splitting the sample to go into a reverse phase to take part of the samples to a reverse phase platform, and then another part to normal phase chromatography. And then within the samples there are phase separation. So we couldn't use this for our purpose. So which is why we wanted to detect, which is why we wanted to come up with a different method. So in short, we wanted to milk the cow, and we have a complicated problem. And what we really wanted to do was to do a simple one like this, crude but direct. That is our approach. What did we do? So we wanted to co-extract all these lipids in a single extraction solvent. And to do that, we wanted to avoid multiphase, because we actually tried to streamline the previous method that I showed, but failed miserably. And so we went back to the board and decided to think about it from scratch. So what we thought was that if you could create a protic environment within a nonpolar system, then perhaps we can extract both polar and nonpolar lipids in the same solvent system. So we explored different solvents, chloroform water, MTB, IPA, and these are solvents that we typically have in the clinical lab. And traditionally, the most common method for extraction involves chloroform, methanol, and water. And it was simply not the right tools for us to work with. So what we found was that a combination of MTB, IPA, and methanol in a ratio of 5:1:4 would provide us with efficient extraction of all the lipids that we wanted to look at. And all right. So what we wanted to do was a one-step protein precipitation, and that's how we do things in clinical lab whenever possible, and that's where we wanted to go. So now that we have an extraction technique that allows us to co-extract all these lipids, we wanted to have a chromatography system that would allow all of these lipids to be retained and eluted the way we want. So we did some method development, not a whole lot. We found that we could use Polaris amide C18 to adequately retain and elute all the lipids of interest. And we also found that we could use a single synthetic ceramide internal standard to reasonably normalize all the signals. This was very useful to us. And also the column allowed us to get almost 3,000 injections before we changed the column. So all these factors were very useful to us in the clinical lab. Perhaps not very important to you in the discovery and the pharma industry. So for mass spectrometry, we wanted to do triple quad mass spectrometry again, and we wanted to look at our eluents using MRM mass spectrometry. We could, in principle, do parent ion mass spectrometry, in which case we would actually get wider coverage, but we chose to do MRM mass spectrometry because of the sensitivity. So the phosphatidylcholines and sphingomyelins, they fragment to a very prominent head group of 184, and ceramides, they fragment to a prominent head group of 264. So this was a clear case where we could build all our transitions around these fragments. Now again, we could use, if you wanted wider coverage, then we would have used parent ion scan. We could use that with this type of fragmentation. So our simplified method, therefore, is that we extract using the formulation that I mentioned. Then we use HPLC built around amide C18 Polaris chromatography system, chromatography column, and then MRM transitions for mass spectrometry. So I mentioned about compiling a large database of transitions. So we ended up with about 271 transitions that we could detect from plasma and plasma HDL and erythrocytes. So we used 271 transitions. But in the discovery mode, where we want to look at as many lipids as possible, we would have, in this case, we would have had to use 3 injections because we couldn't get all 271 transitions into one method. But in the clinical lab, we would actually be using only one of them. So this is the method in picture. So you simply take the extraction solvent, vortex it, centrifuge it, take the supernatant, analyze by mass spectrometry. And this is an example of the data, the raw data that we get. So what this is a total line chromatogram profile for glycated ceramides for 5 monkey samples. So these are samples drawn from monkeys before and after death. So the pre-mortem samples, as you can see, 1, 2, 3, 4, 5, they have significantly different profiles, whereas the post-mortem samples, in the post-mortem samples, this difference is retained. In other words, there is significant difference between patients, but very little difference within patients. So this is the type of information that we look for in the clinical lab. And a similar trend is observed in sphingomyelins, phosphatidylcholines, and ceramides. So now we have everything together. We want to, we have a method. We have, we can get raw data. Now we want to analyze. Data analysis is obviously the most difficult part, and we couldn't do Photoshop. So we had to look for a way to analyze all this data. It's a huge amount of data, 271 transitions. Every sample that every injection that, well, not every sample that we look at. So we accumulate large amount of data, and during the method development stage, we have no idea what the retention time is. So we want a software that would intelligently pick peaks without a pre-assigned retention time. And we couldn't do this with our vendor software. So we had to, again, scratch our heads and come up with, we came up with Skyline. I'm sure there are other approaches out there, but we thought of Skyline because we also do proteomics, and we are familiar with the software. But Skyline is designed for protein analysis. So, but we are from University of Washington, and the authors of Skyline are from University of Washington. So we colluded, and we were given, we were told how to trick Skyline into thinking that it's peptides. So Skyline detects the transitions using a constant part and a variable part. The constant part is where it detects the fragments, and so we assigned, we used the modified peptide feature and told Skyline that the fragments we are looking at are actually modified glycine residues. So we gave it a fake molecular formula. And similarly, we did this for variable part, where it looks for the parents, all 271. So one of my colleagues had to sit there and assign and find out, lie to each of the 271 parents about what their mass should be. So this way we were able, this way Skyline was able to now look at identify our peaks. And this is an example of what throughput. So this is the data from a large number of HDL samples, where we looked at within day CV and between day CVs for a large number of lipids. So the vendor software that we were struggling with would have taken us about 15 hours to do this, whereas with Skyline, we were able to do it in about 4 hours. So that significantly accelerated our efforts. So now that we have a method in place and we have a way to look at the data, the next question was, can we characterize, can we apply this to clinical lab? So characterization of the clinical characterization of the method is actually something somewhat unique to clinical lab, something in the discovery outside you may not be involved in these things. But nevertheless, all your discoveries eventually will end up as biomarkers in clinical lab, and this is the type of work that we have to do. So we looked at the within day, between day, and total CVs. Then we looked at the clinical imprecision where we determined the within patient and between patient CVs and the total. Correlation between patients across multiple days. We also looked at matrix effect, which is an extremely difficult problem to solve in mass spectrometry, but it is very important, especially in clinical lab. So to do this, we looked at ion suppression and ionization efficiencies, and we also looked at the recovery across 5 days for spiked samples to get a feel for how our day-to-day lab variations. So I'm going to show you that data reasonably fast. So these are so this is the imprecision data for a pooled patient sample in plasma, erythrocyte, and HDL. And we detected, in this particular sample, we detected about 228 of those 271 peaks in plasma and erythrocyte. And what we have shown here is the within run and between run, median of the within run and between run CVs for the total lipids and only those lipids that had a between run precision of less than 20%. So what is important here is the total precision, and so total precision is derived from within run and between run, and we got, as you can see, somewhere between 11 to 18% total imprecision, which was acceptable. And similarly, the within and between patient variations. For this, we employed samples from 3 patients drawn at 3 different times and analyzed in triplicates in 3 different days. And we found the mean total imprecision to be 17 to 15%. This was the result of correlation. We subjected 9 patient samples for correlation across 2 days, and we found that the correlation correlation is influenced by how sensitive the peak is. So when we have good peak, we have good correlation. Matrix effect, again, as I want to speed up a little bit. So matrix effect study we did using pre-extraction addition and post-extraction addition. We would split the samples, and one aliquot we would pre-extract, and another aliquot we would extract and then spike standards. And we would compare them with appropriate controls. And this would allow us to obtain extraction efficiencies and ion suppression or enhancement. So here is the data for the extraction efficiencies, where we had somewhere between 80 to 100% extraction efficiency, whereas about 50 to 100% ion suppression or enhancement percentage. So this is something that we have to keep in mind when we are analyzing the data. Then we also wanted to look at the recovery and the CVs of the recovery. So we spiked 4 standards. 4 levels of standards in HDL mixture and used it as a calibration curve and measured the concentration of an unknown, which is a spike target. And then we did this 5 days to determine our CVs. And so we found that the CVs were anywhere between slightly under 20. Now remember, these are concentrations slightly above the endogenous levels. So these are not high concentrations. So we wanted to know whether we can actually detect very low levels, very low increments in concentration. So that is this. And recoveries were anywhere between 70 to 100%. Now that we have a method that we think somewhat confident of applying in the clinical lab, so we brought in our samples, a small subset of it. So we did N of 10 of samples from those patients that were brought to the ER for acute fibrillation and went on to die of myocardial infarction. And then we got 10 samples from patients who were brought with the same clinical presentation but did not, but were discharged and did not have myocardial infarction. And then we compared 10 normal patients. And as you can see here, the ceramide 16 and SM 16 were significantly elevated in patients that went on to have sudden cardiac death compared to the other 2 populations, and with significant statistical, with statistical significance. So, so our next step is obviously to take our large data set, and hopefully something like this might allow us to bring a test the hospital to predict myocardial infarction. Currently, none exists, and it's a huge problem. And with that, I would like to thank the PI, our PI, and Dr. Andy Hufnagel, and then Drs. Roseanne Lemaitre and David Siskowicz. They are from University of Washington School of Medicine. Kim and Jennifer, they are technologists with Golden Hands who helped with many of this work. And Brendan McLean is the author of Skyline, who helped us trick his own baby. So thank you very much, and I would like to...
question for you. Obviously, you said you looked at retrospective samples, so those samples had been stored for a long time. Did you look at the stability of these lipids over time and upon freeze thaw?
That is an ongoing question. We have to. These are frozen in -80 across. So our assumption, our hope is that the freeze thaw cycles wouldn't be different. But that's something that we'll look at in the larger sample. So these 10 samples that we selected were somewhat from different times in the...
Any other question?
As far as I understood, you have not been using scheduled MRMs. Scheduled MRMs.
No, these are just traditional MRMs.
Oh, I got it. So scheduled MRMs will allow you around about 400 MRMs at a time.
Yes, that's a good point. One of the things is that I didn't show the chromatography, but we have, in our system, we have all of them within about 2 to 3 minutes. They all elute in about 2 to 3 minutes. That's also the reason that our internal standards seem to be doing a very good job of normalizing them. So when we try scheduled MRMs, scheduled MRMs come with its own little time lag, and so we found this to be easier. But yes, if you could put them all in one, that would be great. Right, okay. Well, if you had good chromatography...
which is not going from triple quad, but from triple top.
Right. Yeah, and some of the newer systems will actually allow the dynamic... and that would actually solve this problem. Unfortunately, this was not done in that platform.